Now, we are towards the last part of the chapter on photosynthesis, and in this particular chapter, we are just going to focus on one quite important experiment. And this experiment is talking about how we measure the rate of light-dependent reaction using redox indicators. Before we talk about this, I want to do a little bit of revision in chapter 12. I want to do a little bit of revision, and this is linked to the chapter on respiration. If you remember, I'm going to link the video on the top right corner over there. There was one experiment where we used something known as redox indicators to measure the rate of glycolysis, where the glucose, when it undergoes glycolysis during respiration here, it produces pyruvate and it also produces 4 ATP molecules, and it releases hydrogen. And the hydrogen under normal circumstances will be accepted by a hydrogen carrier called NAD, and the NAD will be converted into reduced NAD. Now, we cannot visualize this process happening inside a test tube because, we cannot see the NADs becoming reduced NADs. So one solution to this problem was we used something called a redox indicator. So the redox indicator that we used in the respiration experiment was known as DCPIP. And the DCPIP is an artificial hydrogen acceptor, also known as a redox indicator, and the color of the DCPIP was blue. When it receives the hydrogen during glycolysis, the DCPIP becomes reduced DCPIP, which turns colorless. And in the test tube started out as a blue solution due to the DCPIP, and as glycolysis continued happening, the DCPIP becomes colorless, and that's how you know that the reaction was taking place. Now, we can also use the same concept to measure light-dependent reaction, because in light-dependent reaction, you see, normally we have the photosystem 2, photosystem 1 and the enzyme there. The light would usually hit photosystem 2 and photosystem 1, as you can see there, during light-dependent reaction, and electrons will start moving in a non-cyclical manner. And the electron will eventually reach the enzyme, where this will help to produce reduced NADP and also ATP. But can we see this process happening inside the test tube? We can't. Well, you can actually, because you can actually see the amount of oxygen being produced during light-dependent reaction. But we want a slightly different way to measure light-dependent reaction. You can use DCPIP or methylene blue, where as light hits, and as you can see here, light hits photosystem 2 and photosystem 1. The electrons are moving in a non-cyclical manner. But look at that electron right there. That electron, instead of reaching the enzyme, it will veer off and get accepted by the DCPIP. And when the DCPIP accepts the electron, guess what happens? It becomes reduced DCPIP, where it turns from blue to colorless. So the test tube, if there's no DCPIP, we can't see the reaction taking place. But when there is DCPIP, we can actually see the reaction turn from blue to colorless. And obviously, the faster it becomes colorless, what does that tell you? It tells us that the rate of light-dependent reaction is very fast. As an example, we have two test tubes over here, and if you notice, the test tube is green in color. The reason is because it contains isolated chloroplasts from a particular species of plant. Isolated chloroplasts just means we have removed the chloroplasts from the plants and we have put them in a particular suspended solution, alright, and the chloroplast is, it gives the greenish color. That's why the solution is green. So what we do then is we will then add DCPIP or methylene blue to both the solutions. That's why it becomes blue in color. And in this case, as an example, the test tube at the top, I'm going to give it a low light intensity. That means the strength of the light is not so strong. And the one at the bottom, I'm going to give a high light intensity, which means to say it is stronger light. So obviously in this case, and we will start a stopwatch and we will then measure the time taken for the DCPIP to decolorize. And when the DCPIP totally decolorizes, the blue color will turn colorless and the mixture will return back to its green color, because that's the original color of the solution without the DCPIP anyway. And why did it turn colorless? Well, because DCPIP was receiving the electron during light-dependent reaction. And as you can see here, the test tube at the top took about, let's say, 300 seconds to fully decolorize, but the test tube at the bottom only took about 100 seconds to decolorize. So what does that tell us? It tells us that the test tube at the bottom has a higher rate of light-dependent reaction because the decolorization happened much faster. That's all we have to know about measuring the light-dependent reaction using redox indicators such as DCPIP or methylene blue.